Thank you. So, hello, my name is Bertrand. I'm a postdoc at the University of Sherbrooke, and I'm going to tell you today about our some joint work with Johan Anselmetti, Manuel Lafont, and Aida Wangrawa about the reconstruction of ancestral RNA structures, or more formally, the small parsimony problem on RNA structures. But first, let me start with some primer on structured RNAs. So RNA is mostly known as a messenger RNA, so as a mere intermediate between DNA and proteins. But in fact, as you may know, it also performs directly as RNA a wide variety of other functions. These RNAs are regrouped in what we call non-coding RNAs or functional RNAs. And for these functional RNAs, the folded structure they adopt in space is crucial to their function. So a bit like proteins. Okay? And so it's these folded structures for non-coding RNAs that we're trying to reconstruct ancestral versions of. To be more precise, in this talk, we will handle only what we call non-crossing secondary structures. So these are RNA structures that are in bijection with well-parenthesized strings. So first of all, they are specified by a list of base pairs, so pairings between nucleotides, and any two base pair must either be well nested into one another or side by side, but they cannot cross. Okay, so this gives RNA structures, non-crossing secondary structures, a tree-like structure, which you can draw in the plane like that. And in this drawing, the little regions that are delineated are called loops. And it's these loops that are usually considered as the basic structural elements. So what we'll be looking at is given non-crossing secondary structures at the leaves of a phylogenetic tree, we want to try and reconstruct ancestral versions of these structures. As a database playground, we will use RFAM, which is the main database when it comes to structured RNAs. It contains about 4,000 RNA families, and for each of them, it gives you an alignment, a phylogenetic tree, and some other things. But let me first take a step back about the motivation of this work. So of course, the goal is to try and get some intuition of the mechanisms, the driving forces between RNA evolution and RNA structure evolution, and perhaps on the long term, get some insight into the RNA world hypothesis. But this work also stems from a gap we've identified in the literature, where a lot of work has been done on ancestral sequence reconstruction, but not so much on the structure reconstruction. In fact, we've only identified the two previous articles. This 2009 article, which uses covariance models, if you've heard of them. However, it's arguably quite complex and prohibitive to run, and also restricted as to also restricted as to which structures you can put at the leaves, as to how you do not have complete freedom. And there is this more recent work, but it's not really concerned with structure reconstruction, more with structure constrained sequence reconstruction. Okay, so we thought there was a gap here to be addressed. More formally, we are dealing with the small parsimony problem on RNA structures. So you're given a phylogenetic tree, and at the leaves of this tree, you have labels, which are RNA structures. So given also by these parenthesized strings. And in output, you would like annotations of the ancestral nodes. And the cost function you're optimizing for is the sum of the distances over the edges of the tree. So for each edge, you look at the two annotations at the ends, and you compute their distance and add it. Of course, a crucial concept, a central concept in this definition is the distance function that should be used. And behind that choice is what you consider as your basic unit evolutionary moves in structure evolution. Okay, so a lot of the modeling goes into that. A related problem is a median problem. So you're still given a bunch of structures as input, but this time you're only looking for one structure that has to minimize the average distance to the input structures. It's related to the small parsimony problem because it is sort of contained in it. And some of our algorithms, as I will explain, for small parsimony are based on first the computation of medians. Okay, so as I was saying, in both of these previous definitions, there is a notion of distance that we need to specify. So we worked with 3 different distances. All of them are based on a tree representation of RNA structures. So as I was saying, thanks to the well-nested property of base pairs in non-crossing secondary structures, there is a tree-like shape of the structures. In this tree representation, the leaves are the positions of the sequence, and the black nodes, these internal nodes, are the base pairs. Okay, so a leaf has a black node as an ancestor if it's contained within the base pair that the black node represents. Okay, an important restriction is that we only handle structures with the same number of nucleotides, so we do not handle insertions and deletions yet. And given this tree representation, the 3 distances we worked on are the Robinson-Fultz distance, which is perhaps the most famous example of distance for trees. It's defined as the size of the symmetric difference of the set of clades in the tree, where a clade is the set of leaves that are descendant of a given node. But seen in terms of RNA structures, it's also the symmetric difference of the sets of base pairs. So it's what's called the base pair distance in RNA, in the RNA community. As I will explain, for reasons of resolution of the ancestor that we reconstruct, we will also introduced what we call the internal leaf set distance, in which we compare the sets of internal the sets of internal leaf sets. So an internal leaf set is for each node the set of leaves that are directly attached to it. And in terms of RNA structures, they are related to the loops. So the loops are the regions in between the base pairs, bordered by the base pairs, and they are often considered as the basic structural elements, for instance, in terms of energy of the RNA structures. And finally, as we are working with trees, also a natural candidate for a distance is a tree-edit distance. And for that, we've worked with a relaxed tree-edit distance. Of course, for the details of all these definitions, I redirect you to the paper. To give you some intuition, so the first distance, the Robinson-Fultz distance, so compare the sets of base pairs. So you see here that to go from one structure to the next, you need to remove the red base pair and add the green one. The internal leaf set distance compares the loops, so the regions in between the base pairs. So this loop here is common to the two, but since this base pair here has been shifted, it changes all of the other loops, basically. And the version of the tree-edit distance we've chosen to work with takes into account these shifting moves when it comes to editing the structures. So you see that all of these are like behind all of these distances, there are different choices of what are the unique evolutionary moves through which we think the structure evolves. Okay, so base pair addition, deletion, base pair shifts, etc. Okay. So given this introduction, this was the situation of what was understood of these distances prior to work. And these are our contributions. So of course, given that we introduce the IL distance as a new distance for RNA structure, this column was completely empty before. To give you some intuition of the different results, the small parsimony problem under the RF distance we solve with Fitch's algorithm, or rather, we prove that Fitch's algorithm can be adapted and applied to this setting. For the IL distance, we do not directly solve the small parsimony problem, but we provide an algorithm for the median. And then we are able to apply a median-based heuristic for the small parsimony problem. And finally, as a reference point in our experiments, as you shall see, we apply a distance-based heuristic for the tree-edit distance. I'm going to give you some details of our main algorithmic contributions, this Fitch algorithm and the RNA folding-like algorithm. So the baseline of this first main result is that when you apply Fitch's algorithm to the small parsimony problem under the Robinson full distance with trees at the leaves, then it works. That's basically the main point. The tricky part was to make sure that when you represent within the Fitch setting, you work with vectors of traits, of character traits, so presence of absence of traits with zeros and ones. And here our character traits are base pairs. And so you need to make sure that you reconstruct at the ancestors compatible sets of traits, meaning non-crossing sets of base pairs. And we show in the article that it works. However, an anticipated problem we have with this distance is that we think it, and we show in the experiment that it has a bias towards reconstructing empty structures. Like the higher you go up the tree, the emptier the structures will get. And that doesn't really make sense. There is no reason for the ancestor to have less base pairs than the leaf structure, the structure that's the leaves. And for this reason, we work with the internal leaf set distance, for which we provide an algorithm for the median. Okay, and if you're familiar with the RNA folding algorithm, this algorithm for the median under the internal leaf set distance works a bit similarly. So as I said in the introduction, for the median problem, you're given a bunch of structures. Yes, okay. Sorry for this interruption. Okay. So as I was saying, we provide an algorithm for the median under this internal leaf set distance using dynamic programming over the intervals of the median we are trying to reconstruct. And with this, it unlocks a median-based heuristic for the small parsimony problem. And then what did we do with these results? So we've tried to reconstruct ancestral structures of ORFAM families, so the database I was telling you about at the beginning. And the metric we've looked at, or a quantity we've looked at to assess the quality of our reconstruction is the resolution of the ancestors we reconstruct. And for that, we use a proxy that's simply the number of base pairs. So we would like to be able to make different choices as to how much resolved we want the ancestors to be. Okay. And to get a better resolution, one of the things we do is we also introduce an input loop constraint. So we restrict the ancestors to only have loops or internal leaf sets present in the leaves, in the leaf structures. Okay, so that allows us in this median computation It reduces the complexity of the median computation and also constrains the ancestral structures to look more like the leaf structures. Yeah, so as I was explaining in the beginning, our FAM provides data for a few thousand RNA families. These are structured RNA families or families or sets of putative homologs. So you're given an alignment. It has a consensus structure annotation. So what we first, and it also gives you a phylogenetic tree for that family. So you have a phylogenetic tree with each of the species at the leaves. So the first thing we do is that we completed for each sequence the consensus that is given with the extra base pairs with RNA fold. We've also removed any column with a gap because we do not handle insertions and deletions yet, and restricted the set to a tractable set for our algorithm. And with this set, so we've run our algorithms to reconstruct ancestors. And what we see here in this figure is a representation of the average number of base pairs as you move up the tree. So for height 1, height 2, height 3, etc. So this is the height in the phylogeny. And the color is the average number of base pairs. So this is more for intuitive representation, but what we see here is that we tend to get more blue as we go to the right. So the least resolved technique is the small parsimony under the RF distance, the unconstrained RF distance, as we expected. Then taking the IL distance gives us a little bit better resolution. The tree edit distance gives us even better resolution, but that might be a choice of the heuristic we use for that. And finally, I was telling you about this constraint of the ancestral structures only having input loops or input internal leaf sets. And that gives us a lot more resolution as we go up the tree. So that the ancestral structures look more like the structures at the leaves. Okay. Here is another representation of the same result. So this time we're looking at the number of base pairs at the root of the phylogeny. And we are comparing the choices of metrics and with or without constraint to each other. Okay, so these scatter plots tell you that the RF distance with no constraint gives you less resolution than the IL distance with no constraint. But the RF distance with a constraint gives you more resolution than the IL distance with no constraint, and etc. Okay, and the best resolution are obtained when you put this ILC constraint, input loop constraint, on our algorithms. Finally, this plot represents, so both for our FAM experiment that I have been telling you about, and for random structures, so taking trees and just putting a random structures at the leaves, how the value of the optimal value of the cost function changes when you change the distance or the constraint. And what this graph mainly tells you is that the major driving force behind the differences of value you see is whether you put the constraint or not. So this input loop constraint we have introduced is a strong constraint in the sense of this graph. So yeah, more than the choice of metric, it's whether you put on the constraint or not that you get more resolution in the ancestors or not. So to conclude and take a step back, so we've tried to establish novel foundations for the reconstruction of ancestral RNA structures, trying to fill a gap in the literature that we had identified. The code is available on GitHub. You can put at the leaves any set of secondary structures. And it's really a pure structure focus compared to the literature, which is more focused on sequences. And of course, it's based on ideas drawn from phylogenetics, for instance, a Fitch's algorithm. And it's really a combination of the two, structural RNA bioinformatics and phylogenetics. There is a lot of potential future work that could be done. A relatively major limitation of our work is the fact that we do not take sequence insertions and deletions into account. That forces us to filter our RFAM dataset a lot before doing experiments with it. And so that would be, it would be interesting to take it into account. We still have open algorithmic questions as to the polynomial algorithms for the small parsimony problem or the median. And finally, and it is related to the insertion deletions, we could do experiments across RFAM families, so trying to combine the trees we have for individual families into bigger ones. There is something called RFAM cleanse that starts to do that. But for that, you need to have insertion and deletions because they have different lengths. Thank you.
You remove NCBI. So it's, let's formulate it as a question. When you are using your distances for evolutionary reconstructions, you imply that all moves cost the same, like back and forth. You have two structures and they have a distance, and you can go one way as easily as the opposite way.
Yeah.
So what measure do you find, maybe you investigated, maybe intuitively, what measure better reflects what happens in the evolution? What distances make better evolutionary sense? Because, for example, in the Fitch algorithm, obviously you get a better, an empty set at the root because Fitch algorithm implies that all transitions are equally probable, and they are not. So
Okay.
Any insights from that direction?
So probably you would need to score your evolutionary moves either with some energy values or some sort of proxy for a sort of fitness.
That would be great. But like, for example, Dolo parsimony states that you never gain a character more than once. You can lose it multiple times, but gain only once. And here, like gaining and losing structural elements are not equally not equally unidirectional. So, like, did you look at this from this direction?
So no, we have not wondered about this, but probably this. So if you gain a character and then lose it, I would have to look back at it, but I think Fitch's algorithm does, like an optimal solution to Fitch's algorithm will not feature that.
Yeah, they just optimize the number of, minimize the number of transitions, but they score all transitions equally. And I think actually for future work, this would be the most important thing, finding the directionality.
Okay.
Thinking about directionality of transition.
Interesting. Thank you.
Okay, one more question. Okay. So can you hear this?
Yeah.
So I was sort of curious about an approximation. Your structures are also represented with these parenthesis and dot notation. So they're effectively a binary vector. Did you consider just doing Fitch parsimony on the binary vector, or even ML with a two-state time reversible model or something like that? I don't know if you considered that.
So So under so we sort of already so in here the 0 and 1 representation. So a 1 is for the presence of a specific base pair, so a pair of matching parentheses, and it's really important to keep that matching of parentheses, to keep it in the representation, because also in RNA in general a lot of information comes from that structure. So doing it, I don't know if it's exactly your question, but doing it in a pure sequence way of I don't know, presence of absence or of a character. You will need to make sure to capture this matching.
That makes sense. But are these binary vectors unique for each parenthesis structure? I mean, could you do it on these vectors?
So these are unique. So given all the sets of base pairs you have in all of your input structures, that gives you the length of the vector, and then each structure is present or absent. So these are unique 0 and 1s vectors for each structure.
Thank you.
Yeah.